going to be speaking about cfDNA and ctDNA. What are they and how we measure them? So in every drop of your blood, there are tiny fragments of DNA drifting in the plasma. They come from cells that have died all over your body, and sometimes from cancer cells. These fragments are known as cfDNA, and in the case of cancer, ctDNA. So to understand this further, let's first look at what blood is made of. Blood has two main parts: the cells, or the cellular components, and the plasma. Within the cellular components part, it includes red blood cells that carry oxygen, white blood cells that defend against infection, and platelets that help clotting. Within plasma, the pale yellow liquid that makes up just over half your blood volume, plasma is mostly water, but it also carries proteins, nutrients, hormones, and most importantly, small fragments of DNA floating freely outside of cells. Those free-floating DNA fragments are also called circulating free DNA, or cfDNA. So something important to note is that white blood cells themselves can contain a lot of DNA inside their nuclei. And if they break open after the blood is drawn, they can spill DNA into the plasma and contaminate the sample for which we are looking for the cfDNA of. So that's why we use special collection tubes that stabilize the blood and keep white blood cells intact until the plasma is separated in the lab. So what is cfDNA? cfDNA is a total pool of all these DNA fragments in the plasma. They come from normal cell turnover when healthy cells die naturally, but also from damaged tissue or diseased cells. Because it reflects activity across the whole body, measuring cfDNA is useful in many areas, from prenatal testing to monitoring for organ transplant rejection to detecting disease-related changes. Now you might ask, what is ctDNA? So ctDNA, which stands for circulating tumor DNA, is the smaller category, or is a smaller category within cfDNA. It comes specifically from tumor cells that release DNA when they die or they shed genetic material. And this typically happens from tumors. Tumors will often shed their genetic material into the blood. These fragments carry cancer-specific mutations or chemical changes. Detecting ctDNA can reveal the presence of cancer, track whether treatment is working, and even catch early signs of recurrence, all from a simple blood draw. This approach is also modernly has been coined recently as being called a liquid biopsy. So that's sort of a term that you may have heard on the news lately. So you can think of cfDNA as the entire lake of free-floating DNA in your blood, and inside that lake is a smaller pool known as ctDNA, representing the DNA fragments from tumor cells. So all ctDNA is part of cfDNA, but not all cfDNA will be ctDNA, to put it simply. So how do we measure cfDNA? So measuring starts with a blood draw into a stabilizing tube. The sample is spun in a centrifuge so the plasma separates from the cells. DNA fragments are then extracted from the plasma. So for cfDNA, we measure the total amount of DNA present using methods like qPCR, fluorescent dyes, and bioanalyzers. These are just examples of methods that we could use. Within qPCR, essentially we're amplifying a control gene to estimate total DNA. Within fluorescent dyes, we essentially, the sample will essentially glow in proportion to DNA concentration. And within bioanalyzers, we essentially check both DNA quantity and fragment size. And so naturally, we move on to sort of the next question, which is how would we detect ctDNA? So measuring ctDNA means finding the cancer-specific DNA inside that total pool. So we can search for known mutations using digital droplet PCR or beaming, which can detect a single mutated molecule among thousands of normal ones. We can also use next-generation sequencing to scan for multiple cancer-related changes at once. Or lastly, we can look for sort of tumor-specific methylation patterns, which are chemical tags on DNA that act like fingerprints for certain cancers. So cfDNA is all the DNA fragments. So cfDNA is all the DNA fragments freely floating in your blood plasma. ctDNA is the cancer-derived fraction within it, and by measuring both, we can use a simple blood sample to gain powerful insights from checking organ health to detecting cancer early, as mentioned earlier, without surgery or invasive biopsies. If you like this video, please leave a thumbs up, subscribe, and support us on Patreon so we can continue making videos. Thank you so much.